So I'm Alessandra Traini. I'm working in Peru, and we are hiring. So I want to just show you and share with you how actually I'm using perl since my master's degree, and it helped me to pass from the genomics to the finance where I'm working now. So long story short, I got my master's degree in 2004 when I started working in perl actually. Then my PhD in computational biology because I really love science. And then as bioinformatician, I worked 10 years in genomics. And I ended up in 2015 here in systems working in finance. What is a genome? The nice thing is actually the word genome word really was coined in 1920 in just a publication to connect genes and chromosomes. But actually it became a really discipline just in 1986, 87, comes out in a journal. And this is actually what we are doing now. We are connecting genes and the chromosome. So what is genes doing is trying to collect every microscopic information within a cell, trying to decrypt in chromosome and in the nucleotides that are composing the DNA. They becomes file, big huge file of letters, and we try to decode proteins and metabolics and everything we can find out to actually describe the phenotypic, the disease, the color of hairs, and everything, just starting from the little cell. And I was really lucky because I started a PhD working in the tomato genome project, and it was a huge international project. It was 10 countries. So we got India, Korea, China, Japan, US, UK, Spain, Italy, where I come from, and France and Netherlands. And it was a huge project, and the purpose was to sequence the genome of the tomato as a reference for all the family of tomato. That is a Solanacea. I have 3,000 species in this family, composed by potato, eggplant, pepper, and some ornamental plant. And that's why actually this project was financed by the Heinz for tomato, and actually the genome that is sequenced is the Heinz one. So we actually the sequence potato. And for the tobacco, it was a Philip Morris. And it was really interesting because we start in 2004 with the old generation sequencing Sanger that was really expensive and really, really slow. In 2007, we realized that we didn't have enough time and money to do that, but came out the next generation sequencing that actually helped us to finish in time in 2012. So what is doing next generation sequencing is take all the genome of the plant, is breaking in little pieces and trying to sequence the little pieces. Then the bioinformatics comes out for the assembly when it's trying to order all pieces, put them together, try to overlap, and actually produce the big sequence. It's more clear from here, letters are better. So we actually put together all what they are called reads. We are counting the software, which is the letter for each position. In some cases, we can have 2 letters, could still have a meaning in biology, so we keep it, and we try to order all of them with a different score. So we're going to start with the lowest score assembly, and then we keep going the next assembly on the previous release, try to end up with a good chromosome release. Why perl is really important, why perl is so using bioinformatics, at least when I start. Now they're using but it's a relatively easy language. And if you think that bioinformatics is actually done by biologists, so it's really important. And there are a lot of pre-existing scripts for several purposes and issues, so easy to manage for them. And within the same biolabs, you still have a lot of other scripts and models of already written in perl, and there is still PyPerl, where I'm actually trying to solve everything.
I understood that it was also really important that Perl's regular expression engine was far better optimized than anybody else.
Yeah,
and true. So because of the quantity of data that you're looking at with genomics,
yes, true.
Yeah,
and also it's easy to parse file. The file that usually need really simple, faster file, just each line really easy to parse one perl. And we got a lot of database and did one for genomics. This is an example of NCBI in the US. Actually have everything. From PubMed, that is all the repository of publication. From the nucleotide sequence of the genome, the protein, other expression sequences, the complete genome, other analysis, more or less manual graded, and epigenomics, gene, really everything. Now, it's easy to access by the website or exactly whatever you want. But
Is this free? That's free.
it's free, it's absolutely free. Yeah, you can have human genome in your laptop if you want. Could be useful. But with cPanel, you can actually, for example, with this package, with GenBank, you actually can access to GenBank easily. So with just one line you are in, and you can query for, in this case, is a tech ID, or you can use an access ID, or with a version, because we have several versions of the same gene, more or less annotated, or the GI as a gene ID, or a proper query that could be better one if you can perform on the website. What is really useful? If you think about, we have two point of view. The biologist that needs to write the code, or the bioinformatician trying to write the code for the biology, and you can end up with a code really easy to read and change that someone comes to you every time. This is really useful because it's really easy to read and easy to manage. And then if you think that in genome biology we have a lot of release every, not day, but 3 months. So if you have performed your analysis and you have publication, then you need, your publication is accepted or just on review after 3 months, 6 months sometimes. You need to do it again. So if you are on free, you just download everything and do your analysis, it's great. You don't need to go to ZPB, download, or whatever. Then we got the ensemble one. This is here in Cambridge, is the European database. It's shared between the EPI and the Stanford Institute. It has a lot of genomes in it and a lot of analysis, and also it allows to perform analysis. What I really do is that for each release of the genome annotation, the genome itself, or the analysis, they are releasing the database schema, it's everything public. Database schema, the tutorial, and the perl API. And the database itself. You can really download the database if you want to connect now. So they actually have everything shared. You can connect to their database, normal user. You can download or just get the data you need. And you can perform the analysis in the same way with the same packages they are using in the backend. So you are actually can download their data and re-perform exactly the same analysis that you have on the website. The last I'm going to show you is the software that I use to do all my PhD, second version. And it's GBrowse. It's actually doing what it said. So it's, you can browse the genome. It allows you to set up a website, and you can upload the genome sequence, annotation, and everything you like, or as you prefer. You can actually browse them, and it's actually running through cPanel, and GBrowse itself is packaged there, so as well, you can access through all these packages. You can access to the database, you can just say, I'm interested in this region. Imagine I have a lot of information in this region. Give me this region, on this gene, on this chromosome. It gave me everything that is here, or give me everything that is coding or not coding. Give me everything that is, I don't know, these are SNPs, for example, that would be interesting. So for each one, I can check all the position there, and you can have the nucleotides or the protein in the same region, just using this. And just to conclude, I'm trying to connect what I was doing when I was at university, what I'm doing here, about a perl, but it's actually the system of the data and the flow of the data that we are managing. It's almost the same structure. So there, I've got the university, several users sending me data, several formats, then I'm going to let them to a class And several automatic processes were assembling the data, annotating, clean up, and then there was a manual operation because you need, in biologists, you need human biologists to check the data and improve it. Because there are a lot of prediction of genes or coding sequences of proteins, but you really need to check it. They have an added value when they're checked by a manual operator. And everything was shared on the public website where the user as well from the university can check their own data. What we're doing up here is not something similar. We have the user, our bank, they're sending us their own data. We are not asking for specific format. Send us whatever you have. And with our platform, we are completely automating every process. We are validating, rating, we are reconciling, we are sending out notification inside and outside. We are connecting to the bank itself, sending out notification, we get back response, and everything is shared on a corporate private cloud where they can access to only their own data, and they can constantly check what they send to us. So if they are not sending data, we will come back to them and say, look, you forgot to run the last batch of data, and we do this. And really to and we will go here. And why I will do it again. So I actually respect the university, you think, okay, you was working university is different from a private company. In this case, I would say no, because we are really great teamwork. We are working together. We have 20 different tech with completely different backgrounds, and we are also sharing the office with other 20 different people, and we are constantly helping and teaching each other. Because when I joined, most of us didn't know the business, but you have constantly support by the business, and the business is supported by us for everything that is tech. Try to understand how the system works and how we can help the client. Then we have the client that is continuously challenging us for new tools, new systems, new improvement. And we have the first feedback from them. Then the complexity of the challenge is always bigger, complicated. We get new clients, new big agents, or completely new projects to develop. And then the other thing that I really like when you join here is actually you're allowed to keep your own contribution as soon as you join, because you are absolutely partially free to release. But I mean, if you find a bug, so the first thing they was told to me, okay, you'll probably find a bug if something is not working. If you find a bug, let's fix it. Doesn't matter. If it's a bug, you trust yourself, just do it. And this works really good because you can feel immediately